Our DNA, or deoxyribonucleic acid, is a long molecule that contains our unique genetic code. Like a recipe book, it holds the instructions for building and maintaining our body. The integrity of our DNA faces enormous challenges on a daily basis, from the environment we live in and our dietary habits to our own metabolic processes. Conservative estimates are that more than 10,000 DNA damage events occur per human cell per day. Because DNA is the repository of genetic information, its integrity and stability are essential to life. An assault from the environment and any resulting damage, if not properly repaired, will lead to mutation and possibly disease. Perhaps the two best-known examples linking environmental-induced DNA damage and disease are those of sunlight ultraviolet radiation-related skin cancer, which will be diagnosed in 1 in 5 Americans by the age of 70, and lung cancer related to tobacco smoke, which is linked to 80 to 90% of lung cancer deaths in the U.S. Fortunately, year after year, our DNA remains astonishingly intact due to a host of molecular repair mechanisms that continually proofread our DNA and repair any damage that has occurred. Without these carefully orchestrated repair mechanisms, our genome would collapse. PARP1, or poly-ADP ribose polymerase, uses the molecular tag PAR to mark DNA trouble spots to initiate DNA repair. Unfortunately, methods for studying these repair enzymes in live cells are either limited or have multiple drawbacks. A major need in this field is a reliable, reproducible, high-throughput way to study the effects of these two enzymes and experimental therapeutics targeting them in live cells. Dr. Rob Sobol's research at the USA Mitchell Cancer Institute recognized this need and developed RealPAR, a genetically encoded probe for live cell imaging of PAR. Since PAR is a molecular tag made in response to DNA damage, the ability to visualize in live cells in real time things like when it's made, where it goes and accumulates, and how it's removed are all useful in defining the role of PARP1 and related enzymes in the context of disease, as well as assessing a patient's potential sensitivity to PARP1 or PARG inhibitor therapies. The RealPAR probe is made up of a critical region of the PAR gene, fused with a fluorescent probe that allows for various ways to detect the PAR tag, including microscopy. Importantly, RealPAR can be used in screening of cells for response to toxins in real time, making it particularly useful in high-throughput screening to evaluate the DNA damage impact of environmental or pharmaceutical molecules, as well as in screening possible drug therapies to PARP1 and PARG. High-throughput screening is a high-tech way to expedite the drug discovery process, allowing quick and efficient screening of large compound libraries. RealPAR can also be used to identify patient tumor samples that may be responsive to PARP1 or PARG inhibitor therapy, or that are deficient in DNA repair ability that leads to elevated PAR accumulation, such as the BRCA breast tumor cells. PARP1 has been implicated in numerous cancer types, including breast cancer, ovarian cancer, and colorectal cancer, with PARP1 inhibitors being the therapy of recent clinical trials. But we need to know more in order to develop better therapies. RealPAR does this by enabling live cell imaging of PARP1 and PARG-related DNA damage repair activity, the characterization of genotoxins in real time, the investigation of new PARG inhibitors, and the screening of patients sensitive to PARP inhibitors. As high-throughput screening continues to drive drug discovery, new and innovative ways like RealPAR to assess high-value druggable targets are of increasingly lucrative commercial potential.